The ICAR, National Bureau of Agricultural Insect Resources, or ICAR NBAIR, is one of the 6 national bureaux under the Indian Council of Agricultural Research and is the only research center devoted to advanced research on agricultural insects and allied organisms in the country. This Bengaluru-based bureau prides itself on having one of the best sets of scientists as well as best-in-class equipment for outstanding research in entomology and related fields.
ICAR NBAIR has gained clear visibility amongst researchers, students, farmers, commercial units, and policy makers due to various factors. We have the largest live and insect-derived resources repository in Asia. We own the National Insect Museum now. Some of our most recent successes have been in identification and management of some of the notorious invasive pests, which include rugospiraling whitefly on coconut and oil palm, and most recently the fall armyworm on maize, utilizing totally non-chemical management practices which include biocontrol compatible technologies and semiochemical based technologies.
Since NBAIR has its origins in the erstwhile Commonwealth Institute of Biological Control, the Indian station of which was established in 1957, the legacy of research and development in biological control has been continuing. Indeed, during its first form as the Project Directorate of Biological Control, the research mandate of the Bureau was confined to biological control, one of the safest and environmentally friendly pest management strategies. Project Directorate was restructured into the National Bureau of Agriculturally Important Insects during the 11th Plan. The scope of the organization widened too. The Bureau's activities are now distributed across the 3 divisions of germplasm collection and characterization, germplasm conservation and utilization, and genomic resources. The Division of Germplasm Collection and Characterization focuses on augmentation of insect germplasm collections and maintenance of national repository. The scientists of this division are engaged in biosystematic study of insects, spiders, and mites using traditional approaches. NBAIR Museum got designated by the Ministry of Environment, Forest and Climate Change as the National Repository for agriculturally important insects. Museum has depositions of primary and secondary types, voucher specimens described from all over the country, including pre-independence era. And at present, we have 1.8 lakh specimens with over 250 type species. This heritage repository is a proud descendant of century-old collections, both native as well as exotic species, dating from the Commonwealth Institute of Biological Control, Indian Station. Online web diagnostic portals or web pages are prepared and maintained by the museum researchers under the NBAIR domain. Presently, we are running 10 major databases. The prime areas of expertise are in taxonomy of insect pests groups, namely aphids, coccids, thrips, root grubs, trunk borers, fruit flies, and weevils. Biosystematic studies on pentatomid and non-insect pests like phytophagous mites are also being undertaken. The Bureau also has experts working on taxonomy of natural enemies such as parasitic and predatory wasps. Taxonomic work of generalist predators like spiders has been undertaken by this Bureau since 2016, and we hold a voluminous collection of spiders from various agro ecosystems. The Division of Germplasm Conservation and Utilization is responsible for the live insect repository, which is the largest in Asia. It holds 132 species of live insects being multiplied year-round. Around 1,500 shipments containing 59 million live insects, including hosts and natural enemies, are being dispatched each year by the Bureau. The quarantine facility at NBAIR, set up in the year 2009, has a fully equipped quarantine block as per the international standards. Since then, several natural enemies have been imported, such as Achirophagus papayae for the management of papaya mealy bug and Quadristicus mendeli for the management of Eucalyptus gall wasp. Recently, a seed-feeding weevil, Smicronix lutelentis, was imported from Australia, and the insect is under quarantine screening for the management of the carrot weed Parthenium hysterophorus. NBAIR has worked towards curtailing invasive pest rugose spiraling whitefly through augmentation and conservation of its potential parasitoid Encarsia guadalupe and predator Dicochrysa aster, and foliar application of entomopathogenic fungus Icaria fumosorosea. Another prime area of research is on searching for cadaver-derived Bacillus thuringiensis strains, screening those strains which can express novel insecticidal activities, and to develop suitable formulations. Liquid formulations of Bacillus thuringiensis have been developed, field tested and released for control of Lepidopteran pests. Pseudomonas fluorescens is one of the important and effective entomobacterial pathogen against insect pests. Under the ICAR NAIP-funded project, a unit of pilot-scale production of biopesticides has been set up and mass production of Pseudomonas fluorescens has been standardized. NBAIR has an extensive collection of entomofungal pathogens being utilized for development of non-chemical pest management strategies. Research on nucleopolyhedrosis viruses has been strengthened with the documentation, standardization of production protocols, and utilization of these entomopathogenic viruses against insect pests. Documentation and characterization of entomopathogenic nematodes has been considered as a potential area of research, which can be utilized to manage economically important insect pests, especially root grubs. This bureau has an excellent Chemoecology laboratory with sophisticated instruments such as electroantenogram, gas chromatography coupled electroantenogram detector, wind tunnels, and olfactometers. Insects use olfactory stimuli to identify and orient towards their mates, host plants, and repellent chemicals. This behavioral plasticity is exploited in the development of semiochemical formulations. ICAR NBAIR has developed nanomatrix for controlled delivery of insect pheromones. The pheromone traps can be used for monitoring and mass trapping of insect pests, a technology that harmoniously blends in IPM. At NBAIR, efforts are on to develop attractants and repellents to manage pests of medical and veterinary importance. Potent botanochemicals have been screened to assess their toxicity and repellents to houseflies. This helps to develop environmentally safe formulations to manage housefly menace. At NBAIR, attempts have been made to identify a virulent bacterial bioagent to manage the larval and adult mosquitoes. NBAIR focuses on developing strategies to conserve pollinators. 2 patches of pollinator gardens with more than 100 different species of flowering plants are maintained to provide year-round pollen and nectar supply to the bee pollinators. Our studies have indicated that the habitats of solitary and social bees can be conserved by providing artificial trap nests and pithy stems for different plants. At NBAIR, the black soldier fly is identified as the best option for bioconversion of organic wastes into a compost and as a protein-rich biomass that can be used as feed supplement in fish and poultry industry. The Bureau, in collaboration with ICAR Central Inland Fisheries Research Institute, Barrackpore, Kolkata, has developed a black soldier fly-based pellet feed for aquaculture. In the division of genomic resources, molecular characterization of insects and its resources are being carried out. The division has the state-of-art facility for insect molecular work related to DNA, RNA, and protein. DNA barcoding is one of the major activities under this division. Species identity has been confirmed through gene sequencing for more than 1,000 agriculturally important insects and related organisms. De novo whole genome sequencing of brinjal shoot and fruit borer, Leucinodus orbonalis, has been successfully completed through Illumina and PacBio platforms. The reference quality assembly constructed here is of immense use for gene function analysis. Supercomputing facility for high-throughput bioinformatics analysis is part of the state-of-art facility. Bioinformatics analysis are being carried out for insect genomes and transcriptomes to perform the assembly, annotation, and pathway prediction of various traits. The division has developed genetically improved strains of natural enemies such as Trichogramma and Chrysoperla zastrovi that could tolerate high pesticide load under field conditions. Genomic Resources Division has also developed true pesticide-susceptible iso-female strains of insects for making genetic crosses and for conducting molecular studies. The microflora associated with the insect gut have the potential to degrade insecticides and could make insects resistant to insecticides. Such microflora is screened and are being evaluated for the degradation of insecticides. Farmer-friendly technologies developed by NBAIR are evaluated through the All India Coordinated Research Project on Biological Control of Crop Pests, which is headed by the Director, NBAIR, as Project Coordinator. AICRP-BC has in the past achieved tremendous success in spreading the biocontrol technologies for management of notorious invasives and chemical pesticide-free management of pests infesting paddy, sugarcane, and horticulture crops. The Institute Technology Management Unit, or ITMU of NBAIR, strongly believes that the validated technologies of the Bureau can reach the farmers only if they are taken up by commercial units, for which Institute industry meets are regularly organized. The 34 NBAIR technologies are primarily based on utilization of insects and insect-derived resources through commercialization of technologies to 64 licensees. The Bureau has generated a revenue of more than 1 crore Indian rupees in the last few years. ITMU also manages the IP portfolios of the Institute. So far, 22 patents have been filed and 4 patents have been granted. The Project Monitoring and Evaluation Cell, or the PME cell, of this Bureau is ably involved in framing and research priorities and monitoring the progress of projects by conducting regular meetings such as IRC, RAC, and QRT. In order to create awareness on sustainable agriculture and spread the message on conservation and utilization of insects and insect-derived resources, the Bureau organizes capacity-building programs For researchers, department officials, students and farmers from different parts of the country, the expert taxonomists at NBAIR train researchers on identification of both harmful and useful insects. Agricultural Knowledge Management Unit (AKMU) of NBAIR is responsible for the maintenance of the institute website, e-office, data analysis, high-performance supercomputing facility. Focus is also on carrying out molecular docking studies and developing farmer-friendly mobile apps on feasible non-chemical modes of pest management. NBAIR Library has a collection of 2,188 books, 934 bound volumes, and 2,145 reprints, IP-based access to online databases focusing on agricultural entomology and biological control. The research and other activities of the Bureau are being supported immensely by the technical and supporting staff who assist in the laboratories and various important mandatory programs. The administration and finance wing of NBAIR provides strong, perfect and dependable support to the director in managing the administration. The Society for Biocontrol Advancement (SBA) started functioning from 1996 at NBAIR. SBA publishes quarterly open access online journal devoted to the science of biological control. The Society regularly organizes national and international seminars and conferences on biological control. This ISO 9001-2015 certified organization is the recipient of the Sardar Patel Outstanding ICAR Institution Award, twice in 1998 and 2015. ICAR NBAIR is working on its committed mandate and has emerged as a successful forerunner in the fields of entomology.
I would like to state with conviction that ICAR NBAIR stands for harnessing the power of insects.